Hi guys, Dr. B here. So today we're going to talk about chapter 11, and that's gene technology. So what is genetic engineering? It's the manipulating of genes for practical purposes. Kind of bit of brief history. 1973, Stanley Cohen and Herbert Boyer, they isolated a gene that codes for ribosomal RNA from the DNA of an African clawed frog and inserted it into E. coli DNA. And what happened was that transcription actually occurred, and the bacteria ended up producing frog ribosomal RNA. So now just getting into the basic steps of genetic engineering. First one is cutting DNA, and to do that you use restriction enzymes. They cut out the gene of interest. You also cut out the DNA from the vector. A vector is what's going to be used to carry the gene of interest into another cell. So it can be viruses, yeast, or plasmids. Plasmids refer to circular DNA molecules that replicate independently of the main chromosomes of bacteria. So those are found in bacteria. And then you make recombinant DNA. You use DNA ligase, and it bonds the ends of DNA fragments together. So you'll notice that a lot of the steps involve enzymes. So what does that mean? You probably need to know them. Cloning. Many copies of the gene of interest are made each time the host cell reproduces. So remember, how does bacteria usually reproduce? And it's two words, BF. So binary fission. When a bacterial cell replicates its own DNA, its plasmid DNA also gets replicated. The fourth step is called screening, and this refers to cells that have received a particular gene of interest are separated from the cells that did not take up the vector. So what are some practical applications of genetic engineering? We've talked about two of these actually. The third one you actually kind of know about because you've seen CSI episodes. But the first two are the ones we're going to focus. Well, I think the first three we're going to focus on those right now. Human Genome Project, where we talked about that, BRCA1, BRCA2, TP53. Extracting DNA for a karyotype. Remember, what is a karyotype? It is a way to examine someone's chromosomes. What does this chromosome show? So you look at all of them, hopefully you recognize what's going on here. I'll give you all 5 seconds. 1, 1000, 2, 1000, 3, 1000, 4, 1000, 5, 1000. Okay, time's up. So you look, oh, look at this. There is a trisomy on 21. So this is a Down syndrome patient. And if you look at the sex chromosomes, it is a female because there's 2 X's. Next, what does this chromosome show? What do you notice, hopefully, right here. So this is we talked about 47XXY, now there's 47YY. XYY, I'm sorry. So this is a super male. We talked about the other one before, we'll talk about that in a minute. Non-disjunction during meiosis, that's when this occurs. You have increased growth velocity from early childhood, and they're expected to be a little bit taller than normal. Slightly lower IQ, have some learning difficulties, but fertility and sexual development is normal. What about this one? That's the 47XXX super female karyotype. Similar to the other one, non-disjunction during meiosis. You get a tall stature, poor muscle tone, curve in the 5th finger towards the 4th. You get a small head, microcephaly, maybe some delayed language development, and also a lower IQ. Extracting DNA for forensics. So what forensics usually does is they actually conduct or do something called electrophoresis. And what that means is you're separating out chemicals and you're kind of using electricity to do it. So some chemicals can be blood proteins, DNA, and also inorganic ions. And what's going to happen is that when these things separate out, it's usually based on differences in size or their charge, either positive or negative, or maybe sometimes the smaller molecules will travel further, etc. So what's usually used to kind of run, quote unquote, run the gel is something called a agarose or polyacrylamide gel. So basically this gel is where you put your samples and then you apply an electric current, and that the current causes the molecules to move and separate. It is used in forensic science because it can isolate and compare DNA, which we will do later on, and not necessarily in the whole dramatic sense, but you're going to see how electrophoresis can be used to compare DNA, blood proteins, and other inorganic substances related to the crime scene or suspects. So this is just showing you what it looks like, the contents of it. And you will see that you're going to put the gel in here and the different samples, and here is where the current is going to go kind of quote unquote run through, and then it's going to cause the contents to separate out. So if you look over here on this side, this is, I want to say it's your control because this is called your control, but this is where you can compare the size to knowns. And if you look, this doesn't really mean too much, but if you can kind of see that these are just different genes that are separating out. However, if you look here, let's say this is DNA or blood or saliva found at a crime scene, and you have different suspects that you think may be responsible or may have been at the crime scene, and you take their DNA, and then if you noticed, the only one that really matches up perfectly is suspect number 3. That's how electrophoresis can work for forensics. So that is it for now for this part of chapter 11. We are going to continue the next day, so in the next video. So please follow along, and if you have any questions, let me know.